You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is August 4th, 2025. Our topic is Aerobiology, Air Sampling for Pollen and Spores, presented by Dr. Charles Barnes.
Good morning, everybody. It's Chris Miller. I'm the program director at Children's Mercy, University of Missouri, Kansas City. This is our first and only COLA presentation for today, which is August 4th. We have Dr. Charles Barnes here. Charlie had been with Children's Mercy for many decades and was very instrumental in our aerobiology program, our pollen and mold counting, endless publications. I think we have about 30 years of pollen data here, so pollen and mold. So very instrumental, worked a lot with our fellows through the years. He's always so kind to come back yearly and give an aerobiology talk coming out of retirement. We appreciate that you're here. So with that, Charlie, I'll hand it over to you.
Okay, yeah, thank you very much. And aerobiology is one of those things you just can't escape, you know?
Very true.
Okay, so we're going to talk about today how you take air samples, and these are if you see the pollen count on your local TV screen or go to one of the websites that generate pollen counts, you will find numbers, and we're going to talk about how those numbers are generated and how the pollen is actually collected. And we're going to talk about both outdoor and indoor. We're going to do indoor last, and so if we don't get to it, we'll at least cover most of the outdoor, which is the main area of aerobiology that people deal with. And then we're going to look at the major indoor and outdoor allergens here in Kansas City. These are pollen and spore allergens, and so this is for the Kansas City area. If you're from another area, the importance of these will maybe change a little bit, but this is the main section the main lineup for most of the country. You know, over out in California, you get some unusual ones like eucalyptus, and down in Florida, you get lots of invasive species coming in from South America. But for most of the country, the ones we'll talk about will be the major aeroallergens, aeropollen and spore allergens that you'll see. Aerobiology is something that you probably haven't heard of a whole lot, but it's around and sort of all over the place. NASA has an aerobiology lab, at least they did as of a year ago. Our atmosphere is, and they say that, you know, to justify their existence, they say the answers to life are up in the air. So our atmosphere is home to a wide diversity of microorganisms. And this is bacteria, virus, and fungi. And these tiny life forms hold the clues to big questions, such as where and how life evolved, and if it exists elsewhere. So an aerobiologist is a person that studies these organisms, these particles of biological origin that you find in the air. And they float around our planet's atmosphere. The NASA has actually done some upper atmosphere studies, and they find spores at elevations you would not believe. And pretty much all around the world, the spores you find are the same. And so we exchange spores with Africa. They get big dust storms in Africa. Those storms blow over to Central and South America. The storms from, you know, the aeroparticles from Central and South America on a big low pressure or high pressure system get distributed up here in North America. So pretty much the spores especially are about the same no matter where you are in the world. There are lots of fields that depend on aerobiology. You're used to the biomedical field, but You get respiratory and pediatric medicine, has a lot to do with what's in the air, what people breathe. Environmental health, urban studies, meteorological studies, in lots of places in the world, including the U.S. The pollen and spore concentrations in the atmosphere are part of the weather report. And then you get biochemistry and molecular biology looking for particular allergens and sequencing allergens in the air. And of course, clinical immunology, and the field of allergy is highly dependent on aerobiology. Under respiratory and pediatric medicine, I would add infectious disease in there. I used to go to meetings for companies that make disinfectants, big companies that make disinfectants. And the meetings would be mostly populated by infectious disease people who were studying things like disease transmission in prisons and MRSA, methicillin-resistant organisms and things like that. And I would argue for viruses and bacteria in an airborne setting, and I would lose every time because it was well known by every infectious disease person that it was the fomite that caused most of the transmission. In other words, you put your hand on a surface, you leave the germ there. Somebody else comes along, puts their hand on that thing, they pick up the germ, and then they scratch their nose and put it in their nose, and that's how they get sick. But I think with the most recent pandemic we have had, people are starting to appreciate a lot more the transmission of viruses purely by aerosol means. And there are ways of collecting those, although the military is doing it. I don't know that anybody is doing it on the civilian side. But this is mostly what you as allergists are interested in, and we did this study for 1 year just as we started to collect spores and pollen on the roofs of the hospital in about 1995. And so you'll notice that, and at that time we could easily get statistics on ER visits. All I had to do was, somebody was keeping them in the computer, and all I had to do was ask, and they sent them to me. And wasn't much paper, red tape to go through, and so we're measuring our pollen and we're measuring our spores in blue, and you can see that the spore count starts early in the spring and ends up highest around middle of September or so. And the ER, you'll also notice, goes along in the spring, and it's about, it was about 10 or so, average 4 or 5 a day for respiratory stuff. And then all of a sudden around September, things would spike, and that was a very predictable spike that occurred every September. And of course, there are lots of things going on in September besides the spore count. Ragweed pollen is starting to hit right around the 1st of September. It starts about mid-August, peaks around the first couple of weeks in September, and then drops off. But also the kids are going back to school, they're getting more viruses, so there are lots of things going on. But this just illustrates that you are interested in the aerobiology, especially when it comes to predicting how many people might be showing up in the clinic or in the emergency room at any one particular time. And this I saw in Texas last year. I go to Texas every winter to get out of the cold here. And I saw this on the shelf in the grocery store. And in HEB, as a matter of fact. And if you know Texas, you'll know HEB. And this is basically cedar pollen extract. And they have what's known as cedar fever or cedar fever season down there in Texas. And they get it from all the juniper and cupreaceae plants pollinating in the Edwards Plateau, which is just north of Dallas and stretches on into southern Oklahoma a little bit. And it's really, the cedar trees, you just see them all over the place down there. They've really sort of taken over the Edwards Plateau, and they get extremely high pollen counts. And depending on how the wind is blowing, you get a high pollen count in Texas, and the wind will blow it all the way up here into Kansas City. And this doesn't occur in September. It occurs a lot of times in late December, early January. The cedar pollen doesn't care what day of the year it is. All it cares is the temperature right, and is the humidity correct, and we get a little rainfall. And these cedar trees pollinate heavily, and so you get Texas cedar fever. And at the grocery store, you can buy your oral extract drops here. I'm not going to say anything about how effective they might be. So these are the kinds of collectors you would see to use, or you would have to use to collect pollen to determine a pollen count. And the one on the upper right here is pretty much the standard. This is a Burkhardt collector. They are made in England. They have a website. You can buy these, a couple of $1,000 if you want to go the best, the best stuff and all the accessories. You could probably push it up to $5,000 to $10,000 to get one of these going. There used to be one of these on the roof of the hospital. I don't know if it's still there. A kind of a similar device is the one down below it, and that's the Lanzoni device. Both of these are Hurst type spore traps, and I'll show you a diagram in a minute of a Hurst spore trap. The one here in the middle and the lower is a rotorod device, and it's very unique in that this bar here spins. It has two rods on it coated with a silicone grease. As the rods spin, they impact pollen and spores in the air, and spores and pollen stick to the grease, and you take these in and look at them under a microscope, and you produce a count of particles or pollen per cubic meter of air. This is a typical indoor sampler, and these have pretty much gone out of style now, and you use a disposable unit for most people. But this is an indoor air sampler, and you can see the slit on the top, and it just basically draws a vacuum in here, and there's a little slide on a carrier down below that moves, and you can collect several instances on the same slide. And they're very, very efficient, but most people who do indoor counts now have gone to single-use devices. And then this is an completely automated pollen sense, and this is what Children's Mercy is using these days. And you can see the slit here in the middle, and it goes in and impacts a piece of plastic tape that rolls in front of the a microscope inside the unit here, and the microscope takes a picture of it, and this picture is sent to a central computer that uses artificial intelligence to identify these things. And this is a very difficult thing to do, but I think they're getting it pretty good. But anyway, these are the kinds of collectors you can find. This is the Hurst spore trap. It's very simple. Basically, you've got a chamber here where you draw a vacuum. The vacuum pulls air in through an orifice. Since the pressure outside is 14 pounds per square inch or so, no matter how low a vacuum you get, the pressure pushing it through the orifice is always the same. So this is kind of a critical orifice, and so you get a standard amount of air pulled through here per unit time. There's some sort of clock mechanism that pulls a coated slide in front of the slit over time, and so you get impaction of pollen and spores, which are a little heavier than the air. They come flying through this hole at a terminal velocity here and impact right on the slide, whereas everything lighter goes around. Now, of course, this is going to vary, and the efficiency of this is going to vary depending upon the size of the particles. So things like pollen impact very well. Things like big spores like Alternaria impact very well. Little spores like Aspergillus and Penicillium and tiny little ascospores generally don't impact so well. And so some of those go around. And so the efficiency of collection varies a little bit. But this is the kind of the standard device people use. This is what they look like installed. This is the head of the unit that's placed up here. We draw a vacuum in the center. There's a clock mechanism back here that you have to wind. That was one of our major problems that we had with the Burkhardt device that we had in the hospital was people are not used to winding clocks anymore. And so you could overwind this and you could underwind it, but we generally got it right. And it pulls this slide in front of the little slit that's here in the middle. And you produce a collection that looks like this, and you can tell that this is from James Anderson up in Ontario, Canada. Thanks for the pictures, and I'm in touch with Jim every now and then, and see how he's doing collecting up there. And you can see they probably started it in the morning about 6 or 7 o'clock because the collection gets a little denser as the morning rush hour occurs. Then during the day, kind of levels off. Then afternoon, as people start to go home, you get an evening rush hour, and you get more particles in the air. And a lot of these particles will be things like tire particles and just dust coming up. And then overnight, the concentration kind of is normal until you start the next morning and you get another rush hour, something to stir up the atmosphere. And this is a very simple device. You just put that top on here, and it starts to collect. You can have collections, there are 2 styles of collecting heads here. One style collects everything in 24 hours. Another style collects for 7 days if you want to do a long-term collection. A lot of the practices, allergy practices out there that collect pollen and spores and report their numbers will actually use do an 8-day collection, and they'll count maybe 3 or 4 of those days, and they don't do an everyday collection, but some do. And this is what the slit looks like to pull air in. This is what the 1-day collecting head looks like. This is what the 8-day collecting head looks like. You just take a tape that you coat in silicone grease and you wrap it around here and tape it in place, and you start it, and it runs for 8 days. And this is a diagram of air coming in to impact the slide like that. And this is just how to replace the slide, and this is the collection that you get on the 1-day collecting head. Now there are all kinds of collectors, and this is a very unique one that I saw presented a few years ago, and it's really been a few years ago because basically what they're doing here is they're collecting tobacco mosaic virus, and that's when tobacco got federal funding, and so you know that's been a few years ago. But anyway, Dr. Shields here has got an airplane with 4 little units that hold a petri dish, and he would fly this to a certain altitude and open the petri dish, collect for a minute or so, close that petri dish, fly to a different altitude, open the next petri dish, collect for a minute or so. And so he was getting concentrations of tobacco mosaic virus over this field of tobacco. He may be doing the same thing for soybean now because there's a soybean pathogen that floats around the air as a fungus. But this was a big deal in North Carolina at the time. You see they've got the police out and they've got the road stopped for him so he can fly over this tobacco field and take his readings. I've noticed that North Carolina State has taken this website down, but I've still got a picture of it, and I always thought it was interesting. Some of the other devices, I talked about the Rotarod device, is probably the most frequently used. It's really good for pollen. It's not quite so good for spores, but the Aerobiology research laboratories in Canada produces a similar rotor-rod type device, and I know they're still in business. And pollen.com has bought up the rights to produce the rotor-rod device in the U.S., and this address in Plymouth Meeting is still good. And you can look them up, pollen.com, online, and find just about anything you want about them. And so they're the major commercial site that produces pollen and spore counts. And so when you get a pollen and spore count off of the news or national news or something like that, it generally comes from pollen.com. And what they produce are estimates of today's pollen into the future. And so they're taking data from years past and projecting with a little adjustment for weather and other things that nobody knows about, projecting what the pollen count will be in Kansas City at any particular time of the year, and then sending this out. You can go to their website and see their daily projection. They generally are sponsored by one of the allergy over-the-counter drug companies, Claritin, Allegra, things like that. The best numbers are actually actual counts coming from the American Academy of Allergy, Asthma, and Immunology through their National Allergy Bureau. They are real sticklers for how you do this. You have to have a person at the site who is certified to look through a microscope and produce a pollen count. Basically, to produce a pollen count, you just count the number of pollen at any particular time during the day, or several specific times during the day, and do a mathematical calculation about how much pollen is in the air. And it's still just a statistical estimate, but these statistical estimates are based on the actual pollen concentration in the air for the previous 24 hours. Children's Mercy is not really represented in the National Allergy Bureau these days, because Children's Mercy does a pollen count using a pollen sense device, at least when it's working. And the last time I checked, it wasn't working, but it may be up and running now. And so the National Allergy Bureau puts out numbers in concentrations, and they don't give exact concentrations, but they put out numbers as low, medium, or high, low, moderate, or high for trees, weeds, grasses, and mold. And so you can, if you want, you can go to the National Allergy Bureau and pick the pollen counting station, and these are some pollen counting stations. Most of these are individual allergen offices. Some of them are public health departments. The public health department in St. Louis produces a pollen count on a daily basis. And you can get sent to your email every day the pollen count, the mold and spore and molds, trees, grasses, and weed concentrations for the previous 24 hours. And it's a really nice service, and they do a really good job. They certify collectors. They only take pollen counts from Burkhardt devices or rotor-rod devices, and they only take spore counts from Burkhardt devices. So their numbers are about as accurate as you can get. But it's still an estimation. It's an estimation based on very specific methods and certified individuals, but it's still just an estimation. This is the rotor-rod device. A lot of practices have these. If you want to set up a practice and just put out pollen counts, this is probably the most reasonable price-wise you can get into it. I'm not on the committee. So this device counts onto a lucite rod that's coated in silicone grease. And so the numerical calculation that this device employs will involve a circular calculation, which means it'll have pi in it somewhere. And that's sometimes that's a question on your board exams, how to calculate a pollen count. If it's a pollen count involving a rotorod device, the calculation will have pi in it. But other than that, all you have to do to calculate the spores is just make an estimate of the number of spores total in your collection, the amount of air that your collection represents, divide those two, and you get pollen per cubic meter of air, which is what these are all displayed as. And this is what the rotorod sampler looks like up close. Some of them have a retracting head where the rods are placed in a little device that pulls them back into a protected area when they're not spinning. But basically, you take these rods out, you put them on a adapter, you put a stain on it, you put a cover slip on that, and you count, generally run the microscope right down the middle, and you count the number of pollen you have collected. You multiply that by the width of the microscope, the width of the slide that the microscope sees. You get a total number collected. You multiply that by the amount of air the amount of time that this thing ran, which is equivalent to the amount of air that it collected through. These are not sensitive to wind. So the nice thing about a rotorod collection is no matter which way the wind is blowing, the collection hits the same every time. Whereas the Burkhardt devices, you'll notice, remember they had a big fin on them. They are sensitive to wind. And so if you've got a Burkhardt device, theoretically it faces the wind all the time, but if the wind's blowing real hard, your motor speeds up a little bit, and if the wind's blowing is not blowing at all, the motor speeds down a little bit. And so I do think they're a little sensitive to wind, but they're not as sensitive as some of the other devices that are not wind oriented. Okay. And you can run these. This rotor rod was being run upside down because it was only being run for a short period of time. Normally you mount these with the spinning lucite rods on the bottom, so they're a little protected from rain. But if you get a heavy rain and these things get wet and they're spinning in the rain, it just washes everything off. And so they are a little weather sensitive. The Burkhardt devices are not quite so weather sensitive, but if you get a really heavy rain, you'll still get some water in your collecting head, even in the Burkhardt device. And things change a little bit. But basically, it was published fairly early on that the rotorod sampler and the Burkhardt sampler were pretty much equivalent when it comes to larger particles. That's the bigger spores and the bigger spores and the pollens. But the Burkhardt device was definitely better when you talked about the smaller particles. It was more efficient for the particles below 10 microns or so, which is a lot of small spores that you can find. And then we published later on that the Burkhardt and the Allergenco displayed similar characteristics, but the Burkhardt had a little lower count than the Allergenco when absolute particle counts were low, but similar values when absolute particle counts were high. And I think the Allergenco had a little bit better orifice, but they're really kind of not used outdoors because they're facing upwards, and if it rains, you get everything washed out. Okay, so this brings us to the pollen sense, and one of the major differences here. When you're counting spores microscopically, in order to be able to count them, you put on a stain, and this stain has something like either cotton blue in it or basic fuchsin, And so they're blue or purple, and they also have some glycerol in there to make the pollen swell. And sometimes you make the pollen swell so much that it actually breaks the pollen, but this makes it easy to identify, because when you see a little Pac-Man structure like this, you know it's a Cupreaceae pollen. But when you're collecting for the pollen sense with no stain and no osmotic control, everything looks pretty much the same. This is just a filament off of a plant, and then the air is full of lots of filaments that come off plants. But there's some pollen in here, but they're crinkled up, and you really can't identify them. But the computer, at least I can't identify them, I can't tell them apart. But the computer has the ability to look at these things and analyze each one of them, something that would take hours for me to do microscopically, analyze each one of them and determine if they're pollen or if they're spores, and what kind of pollen or spore they are. And so I think it's doing a pretty good job. The object of doing this with a machine is that you don't have to hire a person. Many of the practices that produce pollen spore counts will have maybe a med tech, or maybe a nurse, or maybe the physician, him or herself, who will take the time out of the day to actually count these pollen collections and take the time to keep the collector running. Well, people are expensive, and so theoretically a machine like this can not only produce a pollen count for a lower price, that's maybe questionable, but also it can produce a pollen count every hour of the day, 24/7, if it's running. And so these collect a lot more granular data, data that's really by the hour by hour, pollen by pollen counts. Now, one year we conned 7 people or 6 people into taking an Allergenco counter home with them and bringing us, changing the slide every morning and bringing it into work. And Julie Landit and I collected these slides and stained them and counted them. And so in the pollen data that Children's Mercy has, there is some data from every 4 hours, 24/7 for most of the pollen season. And so we did a couple of publications off that. The most important one was that we sort of debunked this idea that ragweed was always highest at 6 a.m. in the morning. Well, if you're out collecting ragweed in the ragweed field, ragweed probably is highest at 6 a.m. in the morning, or as soon as it dries off and the wind starts to blow the stuff around. But if you're a person living in an urban environment and you're worried about ragweed exposure, your ragweed exposure really isn't highest until about noon. And so those people could go out and run in the morning and not really have a whole lot of difficulty. Okay, so let's go over some pollens by appearance. And what we'll look at here are stained pollens, the way they look like under the microscope. Some of them, sometimes I have magnified them a little better than my pictures are, but all of these things will have an exine or an outer surface, an intine or an inner surface, and then some material in the middle that takes a characteristic shape when they're stained and when they're swollen under high osmotic pressure. And so we'll be able to identify these. And so we're going to go over elm trees, maple trees, cedar trees. Cedar has really moved up in our area. We get a lot more cedar than we did in 1995. Oak trees, mulberry, and then the weeds, sorrel or dock sorrel or sheep sorrel, grass, plantain, ragweed, and chenopod. And ragweed and chenopod haven't really come up yet, but they're just starting to come up about this time of year. So this is elm. Elm is generally our first pollen that comes out, although sometimes if we have a really warm spring, the maples will beat the elms out. But we have a lot of elm trees in our area. They, after Dutch elm disease went through in the early part of the last century, all the old elm trees in Kansas City died, and they replanted them with a Dutch elm resistant oriental elm that produces these little dime-sized paper seeds. But the pollen looks all pretty much the same, and this is swollen and stained with basic fuchsin, and it has 1, 2, 3, 4, 5 pores on it, and they're oriented around the side. That's known as stephanoporate. And their names of this, if you ever take the course at the academy meeting, they will go over the names and the surface characteristics of all of these things. But we're just going to go over them briefly today. They can have 4 to 7, but most of them have 5. The X sign is kind of wiggly or squiggly, undulate, and this is a very characteristic appearance. And so you see this pollen, and you know pretty much that you've got an elm tree. There are lots and lots of elms in Kansas City, and we have lots of elm pollen exposure. The next one is cedar, and I said this is the Pac-Man, because what's happened is this round pollen, under the influence of the osmotic stain that I put on it, has broken open and spilled its inside. So this is the intine, and this is the exine, or outside of the pollen, and very often the exine looks like the old Pac-Man character, and so this one is a favorite one to be able to identify. We have lots of cedar trees in our area. This is a picture of one out in my pasture. Cedar trees are in the Cupreaceae group. They're inapterate, which means they don't have any pores in them. They're spheroid, round. They have a thick exine that's not very flexible because it breaks, and a thick intine because even though it's under the influence of the osmotic pressure of the stain, it hasn't broken yet. And you can actually walk in the certain times of the year, generally early spring, you can walk up to the cedar trees when they start turning brown and then sort of turn yellow, and you can hit one of them and it just fills the air full of pollen. And that's what I've done here. I've thrown a rock in there and taken a picture of it, and you can see there's just a kind of a yellow cloud around this piece of cedar tree, around this cedar tree. Next one is oak. Oak is in the Quercus group. It's tricolporate. That means it has pores, and the pores have slight little slits among them, and you can see this one's oriented so that you're looking at the pores from the side view. The furrows sometimes can rupture, and it has a granular appearance. Of course, we have lots of oak trees in our area. Because the elm trees that they planted in Kansas City produced such a mess and so much pollen, when they urbanized the Kansas side down the roads there in Overland Park, they planted pin oak trees, and they're beautiful pin oak trees. But if you park your car under the pin oak trees at the right time, you'll come out and your whole car is just yellow. There's so much pollen. Oak produces pollen. These are catkins, and each one of these little round things off the catkins is a flower. And each one of these flowers produces lots and lots of pollen. And so oak trees can produce a tremendous amount of pollen, and it's one of the major pollens in our area. Another major pollen in our area is something you don't think of, which is the mulberry tree. Now, this is a really big mulberry tree out in my pasture that I took a picture of. But normally, mulberry is not only in the big trees like this, but about probably 50% of the scrub trees around here are mulberry. Of course, mulberry trees produce mulberries. And so each of these was a flower, and it's been pollinated, and each little seed on here, on this mulberry, had to be pollinated. And so it has to produce lots and lots of pollen. They're tiny little pollen grains. Most of them are diporate, which means they have two pores. They're operculate, means the pore is a characteristic shape, and it has visible onchi, means you can generally see the hole in this, or where the pollen would come out of. Now, mulberry is also in the Moraceae family. The other tree that most people don't pay a whole lot of attention to, unless you go out walking through the woods and get scratched by them, is the osage tree around here. And these trees are pretty much all over the area here. And since they fight off anybody who wants to cut them down by being really hard and really sticky, you find a lot of osage trees around. We count them, and they are in the Morus family, and so we count them as mulberry, but a lot of the mulberry around here is actually osage tree. And then the first weed we'll run into is sorrel, or dock sorrel. You see this weed in the spring. It's generally up by March around here. You can actually eat, this is an edible plant. You can make a sorrel salad. It grows as a nice green leafy vegetable type thing. It's a little bitter. If you I've tasted sorrel, and it tastes a little bitter, but you used to see in Kansas City, in some of the old Italian areas and the old Croatian areas, you would see some of the older women coming out in the spring to actually collect this and pick it and take it home to cook or make a salad out of. It grows tall. It starts like this, and then it grows tall, puts on a seed head, and the seed heads then turn brown, and they're very they don't deteriorate quickly. And so if you've ever gone through Nebraska and visited the Corn Palace, you'll find seed heads of sorrel all over the outside of it. And this is generally Rumex sheep sorrel. It's tricolporate. Again, you have three pores with slits here, and the exon is finely reticulate. And these descriptions are things that you'll learn if you take the course at the academy meeting. And then grass. We are a grass area. We have lots. We live in the plains, and so we have lots of grass around here. Farmers put up a lot of hay. All grass pollen looks the same, whether it's ryegrass or corn. Corn's bigger, ryegrass is smaller, but they all pretty much look the same. They are monoporate. And they percolate. You can see the pore really well. Surface is fine, and sometimes it's granular, because the grains that you see inside the pollen are actually starch granules that carry energy. So the grass pollen can actually carries quite a bit of energy with it in the form of starch. And grasses are things like Timothy, that has the almost a cattail appearance. Fescue, which is what our major grass around here is, tall fescue, and it produces abundant pollen. And then later in the year, and you'll see at this time of year, you get Johnson grass, which produces a very distinct pollen head on it. All of the spring grasses, Timothy, ryegrass, orchard grass, fescue, are kind of grouped in the northern pasture grasses, and they share a lot of allergens. Whereas the Johnson grass and the Bahia grass and the Bermuda grass actually sort of come from a different group, and they will have a few different allergens in them, but still grasses share a lot of allergens together. Another weed is plantain. This is the weed, if you mow your grass and the very next day there's this spear-like thing sticking up, and has little tiny flowers on it. If you look real closely, that's Plantago lanceolata, or English plantain. And these things will, as long as they're not cut down, they will pollinate over weeks. And they start flowering at the bottom, and they flower all the way up the top. And so plantain you can find in the air anywhere from June all the way through August, and sometimes even in September. And it comes in two forms. This is the English plantain, and this is the Plantago americana, or American plantain. And these things can really grow almost anywhere. You know, if you've got a gravel driveway and you wonder what in the world is that plant that's trying to grow in my gravel driveway, it's probably American plantain. These are periporate. In other words, they have multiple pores in them, usually less than 15. It's the Plantago group or family, and they have a pretty characteristic appearance. You can tell. You can generally see 3 or 4 pores on these, and their pores are spread all around the round pollen grain. And then the ragweed. Ragweed is the big one around here. If you just look at the ragweed pollen grain, it looks irritating. These are in the ambrosia or compositae family. They also are trichoporate, although it's sometimes very difficult to find the pores in there. Very often they will look 3-lobed. However, if you look at them under a microscope, they look like they've got 3 lobes on them. They are sub-echinate, which means they've got little short spikes on them. And ragweed comes in 2 forms. Ambrosia trifida, which is the tall ragweed, and when they say tall, they mean tall. This stuff, you see it growing a lot of times in a swampy area along the side of the interstate or something like that. And these things, the giant ragweed can grow 7-8 feet tall with no difficulty at all. Horses and deer will eat this stuff. And it's not nearly as bitter as the short ragweed. Short ragweed has got this very characteristic leaf on it with multi-lobes on it. And the weed actually kind of droops over, or the leaf actually droops over, and that's where the ragweed name comes from. These will still grow 4 or 5 feet tall, but they'll also pollinate underneath your lawnmower setting. And so if you look very carefully at your grass, if you got a weed down there, a really short one with a little yellow flower on it, this could be short ragweed pollinating in your yard, and it has pollinated. It's been cut off by the lawnmower, but it's still now producing pollen. You also see this all over the sides of the roads around this time of year. We're about 10 days away from the typical start of ragweed season, and the season will run from mid to late August all the way into October. Ragweed is, however, temperature sensitive. If the temperature gets below 56 degrees, the ragweed stops pollinating. However, if the next day the temperature is above 56 degrees, the pollination starts again. Until we get a frost, and that'll kill off most of the ragweed. Also, heavy rains tend to suppress the ragweed count a little bit. Rain sort of washes the pollen out of the air. Now, when you find ragweed, you also generally find chenopod or amaranth. And the biologists have been going back and forth with the names, these amaranthus or Chenopodiaceae name, but the pollens all look pretty much the same. It's this soccer ball look that's got periporate, but more than 15 pores to distinguish it from the plantain. Things that have a weed in the name other than ragweed are generally in the Chenopod amaranth family. And this is, some people will call this pigweed. Now, the trouble with these generic names is that the farmers from northern Missouri will call this pigweed, and the farmers from southern Missouri might call it ironweed, and the people will call this lamb's quarters. But they're all part of the Chenopodiaceae, Amaranthaceae group, And one of the weeds you get in there is gooseweed or goosefoot, and Chenopodus is actually Latin for goosefoot. And if you've ever seen a goose swim from below, you've seen pictooes of it maybe, do you notice the foot as it comes back up kind of folds upon itself? Well, this the gooseweed or goosefoot actually looks like that when the leaf flops over. And you find these all over, and they are starting in South America. They've always consumed the seeds of a chenopod that grows down there, and that is now on the shelf, and you can buy it, and it's called kiwa, and or kiowa, and this kiwa has had the hull removed so that it's actually quite nice to eat. You can order that, you can buy it, you can get it frozen in packages. I haven't seen bulk kiwa in the stores yet. And so that's major pollens in our area. The top 10 spores, and this is a mushroom spore here, looking at the top of it. This is a devil's puffball. They go by lots of names. As this matures, it will get a little hole in the top of it, and when a drop of rain hits this, it will send spores puffing out into the atmosphere around this puffball. And so this used to be one of the shots on the intro to Nova, was a picture of a puffball with raindrops hitting it and spores puffing out of it. But the names that you might recall and we'll go over would be Cladosporum, Ascospore, and Ascospore is a big group of spores. Basidiospores, which is another big group of spores. Alternaria, smuts, which are plant pathogens. Epicoccum spores, Pithomyces, rusts, Bipolaris, Dressleria, Helminthosporum, that you really can't tell the difference by looking at them until you watch them germinate. And then Aspergillus and Penicillium, which again you can't tell the difference by looking at the spores. You have to actually look at the fruiting bodies. And so this is Cladosporum. It's a sort of a sausage-shaped spore. Will have at least 2 attachment sites, and so you start to recognize most of these spores by the attachment sites. Now, you do have the big group of ascospores, and there's a real problem with nomenclature in spores. It turns out that spores have 2 different life cycles. One is a sexual life cycle, in which genetic information is shared between 2 organisms, and the second is an asexual life cycle, where no genetic information is shared. So in the asexual life form, so Cladosporum actually can be classified under the ascospores, but this is the asexual form, so that reproduction always occurs in a uniform manner, and you get a recognizable offspring. Cladosporum is recognized specifically because it has at least 2 pores, or 2 attachment sites, one attachment site, and the attachment site is where the next spore grew. And so this spore is attached to this spore is attached to this one. There's an attachment site where they broke apart right here, and you can see a Cladosporum can have 3 or 4 attachment sites on it. And so it's pretty much the only spore that really grows this way and can be recognized by its sausage shape and the number of attachment sites it has. Now, ascospores are different. Ascospores are from the sexual stage, and they are produced 8 at a time in a bag or sac, and ascus is actually the old Latin word for sac. And so ascospores are produced without attachment sites, and so they'll have a continuous sort of membrane around this ascospore, and sometimes they get really small and really hard to see. And so you can see where sometimes it may be missed on your collection, whereas this down here is a cladosporum coming on the same collection. But these ascospore is just a big group of sort of non-identifiable, because since they're from sexual reproduction, they don't all look exactly the same, and so a lot of times it's hard to identify them, and so they're grouped in unspecified ascospores in the counting collection here, groups here. Now, basidiospores are very much the same way, except they are produced on a basidia, which is a stool-like structure. And so you have a structure that on the basidiospore that has an attachment site and a germ pore, or a place where the DNA would escape, or place where the next organism would start to spring from. And since they're grown on a stool-like structure, there's a little crowding on here. And so all basidiospores are a little asymmetric. And so if you draw a line down through here, this will be asymmetric because they were all crowded around a central area, and they didn't have so much room to grow in the middle, but they had room to grow on the outside. And so there are lots of different basidiospores. Mushrooms are classified under basidiospores. You see lots of mushrooms growing right now because we had such a wet summer here. But we get a lot of, basidiospores in the air right now. Some specific names you get are agaricus and ganoderma, but generally these are counted under undifferentiated basidiospores. Some counting stations will differentiate ganoderma, for instance, but mostly they're undifferentiated basidiospores, mushrooms. And then Alternaria. Alternaria seems to have a very specific place in allergy. It's the one instance where high counts have been definitely linked to increased emergency department visits and even some respiratory related deaths when Alternaria counts are high. And that was published in Minnesota from the Mayo Clinic up there in Minnesota, I believe, several years ago, but it has stuck. And so this is Alternaria. It grows in a line where one is attached to another, is attached to another. The individual you see is normally, looks like this, and it has a tail on it or a beak. And it has both longitudinal and transverse septations, and very easy to spot, and a lot of times Alternaria will be the picture that they'll show you on your exams. Now, smut is a plant pathogen. You see smuts all over the place growing on plants. As far as smuts go, you see a lot of smut on corn around here. And then epicoccum is another ascospore that shows sort of granular, sort of soccer ball-like in their asexual stage. Pithomyces is another ascospore, but in the asexual stage, it comes along as kind of an Alternaria without the beak. And if you think these are related, you're right. And they also tend to share quite a few allergens, and so it's not unusual if somebody tests positive to Alternaria, they might also test positive to Pithomyces or epicoccum. Rust is another plant pathogen. You see rust on wheat around here. You get a lot of wheat rust, and so during the wheat harvest, it's not unusual to see moderate counts of wheat rust. And then Aspergillus and Penicillium. This is Aspergillus, and the structure here, the little club-like structure, is an aspergillum. And if you watched them install the last pope, you saw the guy walking down the aisle with the aspergillum, dipping it in the holy water, slinging it from side to side. And so the actual name of the structure here existed before the structure had ever been seen on Aspergillus, but that's where the name comes from. But the spore is out here on the end, and you really can't tell the difference between that and a penicillium spore. And a penicillium, if this is the aspergillum, this is the old Latin word for paintbrush, penicillius. And so penicillium looks kind of like a paintbrush. It has long finger-like projections with the spores being produced on the end of those. And so you really can't, if you saw this spore and this spore, you really can't tell the difference between the two. In order to differentiate, you have to look at the growing organism there. And so that's for the outdoor sampling. Now, indoor sampling is done quite a bit. There's concern about the adverse effects of indoor mold. You probably get people in the clinic talking about they've got mold growing in their environment. There's some good articles on that. Jay wrote some of them, I wrote some of them, and they were from Jackie in Practice a few years ago. There's a series of questions you can ask to determine sort of how much water they've got in the environment. It gives you a good idea about whether mold in the house is something that you have to worry about. As far as whether you can clean it up, first of all, if you've got fungi growing in your house, it's eating your house, and so it can damage your buildings. It can certainly, if you've got a lot of fungi in a house, it's unpleasant to live in, and it causes lots of adverse health effects in sensitive individuals. In other words, if you're sensitive to mold and you're living in a house that's got high spore counts inside, you're going to have some respiratory problems. Sampling methods include lots of things. You can measure them in dust. You can measure them in. You can measure their volatile organic chemicals that they put off. You can measure their gosterol in their outer walls and the glucans, and some of them even produce mycotoxin. The mold controversy, I think it's pretty much gone away. I think most people will agree that if you've got mold in the house, you've got a potential respiratory problem. EPA, however, takes a pragmatic effect. They recommend remediation of all fungus-contaminated buildings. And so if you've got mold in the house and you can see mold growing, you should take care of that. And if it's on drywall, it's probably throughout the drywall, at least in that area, and the drywall needs to be replaced. So, there are companies that do this on a commercial basis, do mold remediation. Some of them are more ethical than others, but you can find ethical companies out there that do mold evaluation and remediation, and they do a pretty good job. Non-viable collection versus viable collection. Outside, we almost always do a non-viable collection, and so the Burkhardt and the Allergenco, they all collect spores, and by the time you stain them, they're pretty much dead. However, you can collect, and you can identify those by direct microscopically. However, you can collect on a petri dish with the culture medium in there, and you can identify things to the species level in that. However, you can't, if you're collecting a petri dish full, in about a minute or so, you get your petri dish completely full of fungus, and so your sampling time is really short, and so you tend to miss a lot of minor pollen. a lot of minor spores in the air. And some of them will grow in a petri dish and some of them won't. Some of them will grow in one particular media and some of them won't. And the ones that grow real fast tend to cover up the ones that grow real slow. But this is an aerosol. This is an Anderson device. This is for viable. This is for non-viable. This is one they used in the space station and to collect spores and bacteria up there and get an evaluation of how contaminated that living area is, and it's starting to get pretty contaminated. You can also, if you suspect workplace contamination, you can put personal samplers on people. You basically just have to put a vacuum pump on them and some device hooked to the vacuum pump that will pull in the spores and collect them. You can analyze this by visual counts or culture methods. And you can do it by enzyme immunoassay. And so you can actually measure individual allergens straight out of the air. And we did this for ragweed a few years ago. You devised a ragweed assay from a polyclonal antibody ragweed assay. And so we measured the ragweed allergen concentration and compared it to the ragweed particle concentration. And so you can see that even though the particles are not there, there can be ragweed in the air. And however, if there are no particles at all, there generally won't be ragweed in the air. And so we always thought this was very interesting data. So our pollen counts are just examples. They're estimated examples of what might be in the air causing people trouble at any one time. Just because the pollen is not in the air, if it is, you don't see it in the air. If it's not that season, it's probably not going to be much. There's probably not going to be much ragweed allergen in the air. But if it is that season, even though your particle counts are not there, there might be quite a bit of ragweed allergen still in the air. Interesting collection. So I think that's the end of my slides. Does anybody have any questions about this?